Hi, and welcome to the second of two webinars celebrating the July 2027, July 2023 focus issue on fruit crops presented by Plant Physiology. Today we have three speakers, and our host is Richard Espley, one of the editors of the focus issue. Our moderator is Aida Maric, one of the assistant features editors at Plant Physiology. And I will let Aida and Richard introduce the speakers. The focus issue is online now at Plant Physiology. You can see the other editors are listed here. It's our largest focus issue yet, so there's clearly a lot of interest in the physiology of fruit crops. Richard will tell you more about it. We will be posting this webinar to our YouTube channel, where you can find all of our other Plant Physiology and Plant Cell and Plantae webinars. We want to thank ASPB members because membership dues allow us to host these webinars. If you're interested in joining as a member, you can use the code PRESENTS10, which will give you 10% off membership dues. Please put your questions for the speakers into the Q&A questions box. If you have difficulty, you can email me. Please check your internet speed or try dialing in if you're having trouble connecting. We will be emailing you information about getting certificates of attendance 24 hours after the webinar for people who attend live. So look for your email. And I will now turn this over to Richard. Thank you, everyone. You're muted.
And that was just a review for the speakers not to do that, just to practice. Thank you, Mary. I'm just going to share my screen. I have a few slides to introduce the webinar, and hopefully you can see that. Thumbs up if you can. Great, thank you, Mary. So yeah, this is the second of these webinars, and I really enjoyed the first one. As Mary said, I'm really pleased about this. This has been the most successful focus issue so far in plant physiology. And I think that says an awful lot about the state of research into fruit crops. I think it's really good. And we've got at least 25 original papers in here, maybe now even 26. We've got 8 updates, news and views, and a topical review. So I think it says a lot about the state of the research. And I just had a look, and I apologize if you're at the last webinar. Some of what I'm going to say is the same. But just to sort of emphasize that point, really, in 2002, I had a look, and there was one plant physiology paper with apple in the title. And last year, in 2022, there were 13, and 13 so far this year, partly due to this focus issue. But it shows you, 20 years ago, it was quite difficult publishing research into fruit crops, and now it's become quite mainstream, and there's very, very good reasons for that. But I don't need to introduce fruit crops to anybody in this audience, but we know that they're very important to us nutritionally and economically, and there is a real demand for high quality fruit produce that's produced sustainably, and that's very important, I think. But we're facing real challenges, or the industry is, with climate change and water shortages and difficulties with labor. But of course, all of that to this audience, it means that we have more opportunities to overcome some of these issues with the science. Fruit crops, traditionally, many of them really difficult to study. Long juvenility, problems with transformation, and a lack of genetic resources meant that it was quite difficult to do mechanistic studies. But recently, particularly in the last 5 to 10 years, huge change in sequencing and genetic resources, and even now some really good new techniques and transformation that are helping those recalcitrant crops. And I think these are particularly important because right now we need to know about the genes that can fix some of the problems that fruit crops and producers are going to face in the future, whether it's by gene editing or some other new breeding technologies. So I these images up last time, a couple of weeks ago, and on the left here you see the lovely cover of Plant Physiology with some beautiful apples from the Hawke's Bay orchard in New Zealand. And then next to it you see these rather troubling photographs which have flooded orchards. These are the same orchards that featured on that picture in Plant Physiology, and that was after the cyclone hit parts of New Zealand in 2023. So just recently, and I think that tells us a lot about what's going on and what we need to do. And of course, whilst these are very damp pictures in other parts of Europe and China and the USA, there's some really high temperatures right now, affecting many of the growing regions, the fruit growing regions around the world. Anyway, getting on to the focus issue. Before we talk about some of the original research, I just wanted to give a plug really for some of the updates, the reviews. There's a really good range of reviews in this focus issue. I've just put up 3 here as an example, cold tolerance in grape, vitamin C metabolism and regulation in fruit, and some interesting new thoughts on epigenetics in climacteric fruit ripening. But a heap of really interesting reviews to look at. But we're really here tonight to celebrate some of the really good original research papers. And we've got papers in this issue on stresses, biotic and abiotic, and phytohormones and pigments, and all sorts of things, transcriptional control and epigenetics. And really interestingly, it wasn't devised this way, but in a really broad range of fruit crops. So it's not just tomato or apple, it's a whole heap of crops that people are doing excellent, high quality research. And just on the right-hand side, we've got some examples of like acid in the middle there, regulation in apple, and sugar regulation in citrus above that, and below in sweet cherry, some new BBX genes that are driving anthocyanin control. So a whole heap of different things. I just put those up as examples. 2 weeks ago, we had excellent talks from Christina, Jing, and Erica on some really interesting topics, and you can go and look at those. There's a recording on YouTube. But tonight is all about Jessica, Qingmei, and Zefeng, and they're going to talk about some of the research that they've published in Plant Physiology, and I'm really excited to hear about that. I'm going to hand over to Aida, who's going to introduce the speakers tonight and moderate this session. So I've got the easy job. But before I go, I just wanted to thank my fellow editors, Lailiang Cheng from Cornell, Fengwang Ma from Northwest, Mickael Malnoy from FEM in Italy, and we were led by Professor Jun Wu from Nanjing. So thanks to all those. It's been great fun doing it, actually, really enjoyed it. Thanks to Mary and the team at Plant Physiology, and of course, if there's anyone out there who did any reviewing, thanks to you, because that's really important. And yeah, I look forward to hearing from Jessica, Qingmei, and Zefeng. Thank you. Aida, over to you.
Thank you very much, Richard. It is my pleasure to introduce our speakers for today. So first of whom is Jessica Trinh. She is a 5th-year PhD candidate in the Gita Corker Lab at the University of California, Davis. And she is looking in the evolution of microbial features in vector-borne bacterial plant pathogens to understand how they interact with the host's immune system. Jessica was recently selected for the 2022-23 Jester Shields Graduate Research Award from the UC Davis to fund her conference travel this year. In addition to the research, Jessica is involved in outreach and science communication programs on campus. So the CV sounds promising. Looking forward to your presentation whenever you're ready, Jessica.
Oh, thank you so much for the introduction. Yeah, I'll go ahead and share my screen. So I'm going to assume that that's not the presenter view, so that's fantastic. So yeah, I mean, I'd like to thank all the organizers for having me here today. It's been a real pleasure to work with all of you. So I'd like to share some of my research now. So when it comes to citrus, like many cultivated crops, oh, sorry, hold on, is it now it's advancing, sorry. So Citrus, like many other cultivated crops, is susceptible to several different types of pathogens. So I've listed fungi and bacteria here, but there's also viruses, oomycetes that are also part of this whole picture. And all these different pathogens cause millions of dollars in diseases worldwide and have a significant impact on our economy. So we really want to try and solve these issues. When we're thinking about the susceptibility of citrus to these pathogens, we have to think about how we breed them. It takes so long to make new cultivars of citrus from traditional breeding alone, so nearly all the cultivars of citrus crops have been either from spontaneous or induced mutations. And what we do with those mutated clones is we actually propagate them asexually by grafting, tissue culture, and various other methods. So these clones just get propagated, which is great if you're trying to retain horticulturally relevant or traits that you want, but not so great if you have a pathogen on the loose and you have a bunch of susceptible citrus. A lot of researchers now are trying to take disease resistance mechanisms from other species and put them into citrus to try and generate more resistant cultivars. And when we think about resistance mechanisms that are compatible with citrus, it's pretty important to look in the neighborhood, the evolutionary neighborhood of where citrus is. So Rutaceae is such a huge family that I've only listed one subfamily, Aurantoideae, right here. Citridae, right here, is where cultivated citrus lives. And but again, it's one subtribe out of all these different subtribes. When you're looking at individual genotypes within different subtribes, you can see that within Clausinae and Balsamocitrinae, for example, you've got genotypes that have different fruits, different branch and leaf morphologies. There's just so much variation across two different subtribes. When you're looking within a subtribe even, just within Citrinae itself, you've got oranges, lemons, tangerines, all this stuff you can just buy at the grocery store. That being said, since we have so many different genotypes within just one subfamily, it kind of poses the question, how do disease resistance mechanisms vary within a family of related genotypes? So one really important disease mechanism that's important for plant defense, disease resistance mechanism that's important for plant defense are these perception of microbial features. So plants have evolved these really sophisticated pattern recognition receptors that are just proteins that sit on the membrane of your plant cell, and these are responsible for detecting microbes, specifically microbial features. So in general, some types of microbial features can come from bacteria through proteins such as flagellin or cold shock protein. These features can be on the surface of the bacteria, such as, again, the flagellum, which is attached to the bacterial cell, or is produced by the bacteria inside the cell, but becomes released when lysed. And these different features that are perceived by plants are known as microbe-associated molecular patterns, MAMPs for short. Most of the time, I'll refer to them as MAMPs. I'll switch back between MAMPs and microbial features, just so y'all know. When it comes to recognizing these microbial features, these MAMPs, they don't have to be a whole protein. These receptors are sophisticated enough to recognize just one epitope of these proteins. This can be around 20 amino acids or so, give or take a few. These will bind to the receptors of these plants and cause them to co-localize with co-receptors, cause some signaling cascades, which will lead to a number of physiological responses, such as reactive oxygen species production, transcriptional reprogramming, and salicylic acid production, which combined kind of create this environment that is hopefully inhospitable for pathogens. So again, we are going back to this question of like how this particular mechanism could vary across related genotypes. If you're looking within a genus, for example, even with just plain old Arabidopsis, you can see that there's a huge variation in the diversity of whether or not the same receptor from that genotype can perceive bacterial flagellin. So you got little genotypes on this one end that are not very sensitive to bacterial flagellin, but on this end, you've got some that are very sensitive, which kind of shows that, again, this is just within one genus. It kind of begs the question that I asked in the beginning, how do these resistance mechanisms vary within a family that has several genera, not just one? So two of the things that I wanted to talk about from our paper was basically our screen testing the ability of the citrus genotypes and their relatives to detect pathogens, and also determining whether this variability in their immune responses is due to the difference in the pattern recognition receptor itself, whether that's important. So again, I've indicated that these Rutaceae genotypes have just so many variability in between them. It's so hard to screen all of them individually. So we developed this high throughput reactive oxygen species assay to perform this very quick evaluation of whether or not something is immunogenic in a particular genotype. This can be done in 96 wells. So again, high throughput, we love high throughput. And all you need is a small piece of leaf. You can add water or the microbial feature of your choice to induce the production of reactive oxygen species. And then you add some chemicals that turn that reactive oxygen species into luminescence that can be read on a plate reader and then graphed onto a plot that shows the production of ROS over a period of time. And this is more or less the bread and butter of how we were able to do a massive screen with 86 different Rutaceae genotypes, again, some citrus and some of their relatives, and we screened them with 3 different pathogen features, 2 of which are from bacteria, such as CSP22, which comes from bacterial cold shock protein, again, as you recall, the particular epitope that the plant recognizes, and Flag22 from bacterial flagellin. We also screened these Rutaceae genotypes with chitin from either fungal cells or from the exoskeleton of insects. They're present in both. And we took all the data that we generated and we plotted the maximum relative light units from each individual ROS curve as a heat map, just to simplify all the data for y'all. So I'm going to show you a heat map, but just note that the more intense the red color, the more strong the reaction is to the particular microbial feature by that genotype. So if we're just looking at citronate, again, which contains cultivated citrus and their cousins, we can see that there's a huge amount of variation in microbial feature perception. If you're looking in that first column right there with chitin, you can see that a vast majority of these genotypes actually do respond to chitin. To varying degrees, sure, but they most respond to chitin. That kind of drops off to about half when you look at cold shock protein. So around here, there's only about half that can see or perceive cold shock protein. One shocking thing that I found was that only 33% of these genotypes, again, which contain citrus, respond to bacterial flagellin, which is in the middle column here. This will come in handy later, so keep that in mind. When you're looking at relatives of these citrinae, again, in different subtribes, like the Clausinae and the Balsamo citrinae, you've got genotypes that can respond to multiple of these maps, but again, we didn't test too many of these genotypes, so make of that as you will. So we did this massive screen with all these genotypes, and we're going to pick apart these a little bit more by looking at the differences in their receptors and seeing whether that has any effect on their immune response variability. So we're going to look at 2 different receptors for the rest of this talk, LIC5, which is chitin sensing, and FLS2, which is flagellin sensing. So as I mentioned before, chitin perception is pretty spread out across the citrus genotypes. And we know from previous studies that the Arabidopsis version of the receptor is pretty important for chitin perception. And that receptor is called LIC5. We wanted to see if this LIC5 receptor was conserved across citrus to see if it was still functional. And we took the LIC5 sequence and we tried to pull it out amongst all the different land plants and saw that if you're looking at the citrus versions of LIC5 right here and there, you can see that they're clustering with other eudicots, which means that they are, well, they're closely related, which makes sense. So we also, I'm not showing this data here, but we also took these citrus LIC5 copies and we also cloned them into Arabidopsis that has LIC5 receptor knocked out and found that no matter whether the genotype was a weak or strong responder of chitin, it can still cause that ROS burst in Arabidopsis. So we're pretty sure that the chitin receptor is an ancient receptor passed down from an ancestral organism that remains conserved in citrus, which is why we have so many citrus genotypes that can perceive chitin. Now, I'm going to tell you about something that's a little less conserved. So remember when I said how 36% of genotypes in citrinae can actually respond to Flag22? Well, we took a little bit of a deeper dive into that, especially in citrus, to see why that might be occurring. So I'm showing you a graph of the individual ROS curves for Lisbon lemon, and Washington navel is in there too. But the thing is, the Washington navel curve is just so small, it's practically negligible. There doesn't seem to be much of a ROS burst there. So we also took a look at the genomes of Lisbon lemon and Washington navel to see whether they may be lacking this receptor or not. Turns out a particular haplotype of Lisbon lemon and Washington navel actually have two different copies of the flagellin receptor. They're in tandem on the same chromosome. And what's really cool is that Jerry Lee, another graduate student in the lab, cloned these receptors into silenced Nicotiana benthamiana, so silenced so that it doesn't perceive Fly22 anymore. And regardless of whether or not the receptor comes from Lisbon lemon or Washington navel, there doesn't seem to be a difference in whether or not the Nicotiana can perceive the Fly22 strongly or not. So again, both these two citrus type receptors can still confer recognition of Fly22, which shows that maybe the difference in the perception between two different genotypes may not just be at the receptor level. It could be in the signaling system that are downstream of that receptor, which remains a mystery. We're still trying to figure that out. So one last thing that I wanted to cover before I wrap this up, which I thought was really interesting, was can we find genotypes that perceive microbial features from relevant citrus pathogens? Now, I told you about two different bacterial ones, cold shock protein and Fly22, that are, they're pretty conserved, but the thing is, we don't know if the sequences are exactly the same as a relevant citrus pathogen one. So for this one, I'm going to talk about Candidase Liberibacter asiaticus, or CLAS for short, which is the causative agent of citrus lawn lumping, one of the most deadly diseases of citrus in human history. And when we compare the two sequences, the one that we screened with our 86 different genotypes and the CLAS version of it, you can see that there's quite a few amino acid differences between these two sequences that may affect whether the plant actually perceives it. And lo and behold, we actually can find several genotypes actually that can perceive the CLAS version of the MAMP when the rest of these genotypes do not perceive a thing. So the one thing to note is that out of the 86, only 7 of them can consistently perceive cold shock protein from CLAS, which kind of highlights the importance of our ROS assay. Without our high throughput assay, I'm not sure if it would be super easy to find these 7. So it'd be really cool to see if these have any particular resistance mechanisms against Liberibacter. So to summarize everything, the perception of microbial features varies. That seems like not too much of a, seems like a no-brainer. These ROS burst assays are very useful for these high throughput screening. You don't need a genome resource. You just need leaves and some chemicals, which is pretty cool. And if we can also use homology, so again, receptors that we know are in Arabidopsis or another model plant that we could take, what we could try and pull out from citrus or other relatives and see if we can try and translate that into susceptible genotypes to make them respond to MAMPs and have them hopefully be more resistant to different pathogens. So with that, I'd like to thank all of you for listening. Here's my contact information if you want it. I'd like to thank all my funding sources and all the people that are in bold that really worked really hard on this project. It's a multi-year study, so it took a while to get this out. If you want to read the whole paper, it's right here. Scan this QR code if you may. And with that, I'd like to take any questions. Thanks so much.
Thank you very much, Jessica. So yeah, if you have any questions, you can feel free to type them down in the chat box. Okay, we already have a question from Faisal. So Faisal is asking, so there are many ROSs, but which ROS did you detect specifically?
It's a really interesting question. So the assay that we use is not very specific, so I don't think it could narrow down this particular species of ROS, but it's a very general burst, and we've tested it with several positive controls to know that it's specific for the immune response. So it's a general ROS detection system, not specific, if that was what you're looking for.
I think Faisal referred to this. And okay, we are still waiting for some additional questions. So maybe in the meantime, you can maybe comment on the future of this research. Like, how do you envision what happens next?
Yeah, so one of the things, I mean, I mentioned this before, but like the genotypes that I looked at earlier, sorry, give me a second. These would be really cool if we could actually get the genome resources from that and be able to pull out novel receptors from those and clone them into citrus. That would be super awesome if you could spend all the money on sequencing. If we can't, then one of the things I would like to look at are downstream signaling mechanisms in citrus that may be broken or something. Like I mentioned before, these genotypes are often mutagenized or spontaneously, like the spontaneous mutations are used for cloning. So they may be some broken downstream component that might actually be really important for immune signaling. We're not sure.
And precisely this is the next question that we had from Vani, who asked, is there any finding in relationship with signaling? This is probably the future. So
future.
question is, how difficult is it to transfer receptors from pathogen-resistant cultivars to pathogen-sensitive ones?
Yeah, so there are ways to transform citrus. Tissue culture technologies are a thing. It just kind of takes a while. Most of it kind of depends on how long you're willing to wait for those genotypes to come back. So I think the difficulty mostly comes from like transforming the citrus, getting those to take, getting the construct to take, and then having those grown out, which can take years because citrus takes forever to grow. So
very good. Thank you so much, Jessica. I think we should move on to our next speaker, but if you have additional questions, I'm sure Jessica will be happy to answer them. So our next speaker is Qingmei Guan. She is a full professor at the Northwest ANF University in China. She received her PhD and postdoc training from the University of Maryland, and currently she's leading a research group whose research interests include molecular mechanisms of plant responses to abiotic stresses such as temperature or drought stress, as well as molecular marker-assisted apple breeding at the whole genome level with stress-resistant traits. So we are looking forward to your talk, Qingmei, whenever you're ready.
Hi, thank you for the introduction. I also want to thank Mary for organizing this webinar. Today, I'm going to share our research stories just published in the Plant Physiology this year. So apple has the largest, and China has the largest apple producing area in the world. However, environmental stresses, including cold stress, can limit the apple production and also the fruit quality. For example, the cold temperatures occurred in the spring can be lethal for the flowers. So like the annual model plants, apple trees also have a signal transduction process, including signal sensing, perception, and transduction. Our lab is focusing on the identification of important players or markers in apple stress response, characterization of these players, and molecular breeding. Previously, we found that two transcription factors, MIB88 and 124, are positive regulators for apple cold stress tolerance. As you can see that the overexpression plants are more tolerant to the cold stress, but the iron plants are more sensitive to the cold stress. As transcription factors, we found that MIB88 and 124 can positively regulate the expression of some cold responsive genes, including CCE1, which is CBF-dependent genes, and also CBF genes and core genes. Because CBF genes are responsive for the anthocyanin accumulation, consistently we found that the MIB iron plants have less anthocyanin accumulation and cold stress, but the overexpression plants have more anthocyanin accumulation. Because anthocyanin usually is responsible for the ROS scavenging, we then detected the ROS level in the MIB88 and 124 transgenic plants, and consistently we found that the overexpression plants have less H2O2, but the RM plants have more H2O2 under cold stress. We further found that MIB88 and 124 can directly bind to the promoter and positively regulate the expression of CBF-dependent genes, including CCE1, and CBF-independent genes, including CSP3. So for this research, we propose that MIB88 and 124 can bind to the promoter of CBF-dependent genes, including CCE1, and CBF-independent genes like CSP3, and positively regulate cold stress tolerance. The CBF-dependent genes, including CCE1, can further promote anthocyanin accumulation and H2O2 detoxification and cold stress. So to further understand the molecular mechanism of MIB88 and 124, we try to screen to identify the interacting proteins of MIB88 and 124 and identify microRNA biogenesis factor HIL1. We found that MIB88 and 124 cannot regulate expression of HIL1 and cold. However, the HIL1 can regulate the accumulation of MIB88 and 124 transcripts and also protein level under cold. Further, the genetic interaction analysis shows that HIL1 is an upstream factor of MIB88 under cold stress. So this data indicates that the HIL1 might regulate cold stress by regulating expression of MIB88 and 124. So using the transgenic plants, we found that HIL1 is a positive regulator for apple cold tolerance because the RNI plants are more sensitive, but the overexpression plants are more tolerant to the cold stress. Because MIB88 can regulate the anthocyanin accumulation and cold stress, we then also detected the anthocyanin level in HIL1 transgenic plants and found that under cold stress, the RNI plants have less anthocyanin accumulation, but the overexpression plants have more anthocyanin and cold stress. Consistently, the HIL1 is a positive regulator for ROS detoxification and cold stress. Because HIL1 is an important player for microRNA biogenesis, we then also performed a small RNA sequencing analysis using the HIL1 RNA plants and cold stress, and found that HIL1 can regulate accumulation, the transcripts of 8 cold responsive microRNAs, including MIR172, MIR160, and MIR156. Previously, we found that MIR160 is a negative regulator for apple cold tolerance. So next, we generated the transgenic KLI of MIB56 and MIR172, and found that the MIR156 is a negative regulator for apple cold tolerance, but the MIR172 is a positive regulator for apple cold tolerance. In addition, we also found that MIR156 and MIR172 are responsible for apple ROS detoxification under cold stress. So for this research, we proposed this working model. So under cold stress, HIL1 can positively regulate apple cold tolerance by regulating the transcripts of MIB88 and 124. In addition, HIL1 can also positively regulate cold tolerance by regulating the transcripts of, or by regulating the biogenesis of cold responsive microRNAs, including 172, MIR156, and MIR160. So to summarize, we found that MIB-ADE and 124 play positive roles in alpha cold stress tolerance, and the interaction protein of MIB-ADE and 124, the higher one, which also play a positive role in cold tolerance of apple by positively regulating expression of MIB-ADE and 124, also by regulating the biogenesis of cold responsive microRNAs, including MIR-160, MIR-156, and also MIR-172. So this work was done by Dr. In my previous PhD students, Dr. Yinpeng Xie and Dr. Xiaoxia Shen, and also my current student, Jieqiang He. And I also want to thank my collaborators and also funding supporters. And also thank you. I'm happy to take questions.
Yes, thank you very much, Qingmei. So again, if you have any questions, our chat box is always open. While we're waiting for a participant's question, maybe if you can comment on the possibility of transferring whether the same mechanism could work in fruits themselves, and if this could affect like post-harvest storage.
I don't know, because I never worked on the post-harvest the chilling stress. Actually, in our lab, we are more focusing on the freezing stress, so which is a temperature less than below 0. I think this should be different with the post-harvest physiology, that chilling stress. So yeah, so the answer is that I don't know. I'm not sure, but I guess should be different.
Okay, let's see if we have any question in the chat box. Okay. So, okay, people are commenting on the fact that, okay, H2O2 is a signaling molecule, so is it important for plant?
Oh yes, this is an interesting question and also very good question. As one of the ROS components, H2O2 is also very important because it can also be a signaling molecule. But under stress, including especially the abiotic stress, including drought stress, cold stress, or high temperature stress, under these kind of stresses, plants will generate excess H2O2. So in this situation, H2O2, I think is not a signaling molecule. So I think it's harmful for the plant growth under stress. Yeah, I'm not sure if I'm clear. H2O2 sometimes is signaling molecule, but under abiotic stress, especially the drought, cold, and high temperature, H2O2 actually is harmful for the plant survival.
I think that answers the question very well. And then what about the MDA lipid peroxide you measure?
Actually, we didn't measure the MDA. We didn't measure that. But I assume that MDA content should be different between the transgenic plants and the wild type, because we did measure the ion leakage of the transgenic plants and the wild type. So assume that the MDA content should be different.
Okay, and just one more last question. So during cold condition, accumulation of reactive oxygen intermediates causes is regular?
Yeah, I think so. Under abiotic stress, as I said before, including cold stress, also called acclimation, the ROS and the ROS accumulation is regular, especially the excess ROS accumulation.
Okay, very good. This sparks a very lively discussion, I guess, but we have to move on to our next speaker. Thank you so much, Qingmei.
Okay, thank you, Aida. I may answer the questions online.
Perfect. Thank you so much. So now we will move on to our last speaker for the day. So it's Zefeng Zhai, a PhD degree recipient from the China Agricultural University. His research is focused on the molecular mechanisms of fruit softening in sweet cherry. He currently works in the Agricultural Genomics Institute in Shenzhen, the Chinese Academy of Agricultural Sciences, as a postdoc, and focuses on the molecular breeding of diploid potato. So a lot of interesting species you're working with, so we're looking forward to your presentation whenever you're ready.
Thank you, Aida. Hello.
I cannot hear you. I think you're muted.
Now it's okay?
Yes, now it's fine. And we can see your slides. Yeah.
Okay. Hello, everyone. So happy to participate in the webinar on frog crops. I'm Zefeng. The content I want to share with you today is my work in China Agricultural University during my doctoral period. The theme of my presentation is abscisic acid responsive transcription factor 2, 6, and 15 mediated fruit softening in sweet cherry. So the first question is why we study the fruit softening in sweet cherry. Sweet cherry is a popular fruit because of its appealing appearance, delicious flavor, rich and nutritional value. However, the ripening process of sweet cherry fruit is accompanied by a rapid decline in firmness, we call softening. The direct consequence of fruit softening is that the fruit is fresh is susceptible to rotting after harvest, strongly limiting the circulation of this fruit to the consumer market. So dissecting the mechanism behind softening is a prerequisite before attempting to modulate the process. So the second question is, what is the physiological causes of fruit softening? Numerous studies have found that cell wall modifications are the predominant causes of fruit softening. The structure of cell wall is like a concrete wall filled by many substances such as cellulose, hemicellulose, and pectin, and various proteins. The cellulose, hemicellulose, and pectin are main polysaccharide composition in the cell wall during the fruit ripening and softening. The cell wall polysaccharides, including cellulose, hemicellulose, and pectin, are hydrolyzed by a set of cell wall modifying enzymes, leading to the loss of cell structure and the reduction of intercellular adhesion. So the third question is, what kind of enzymes or proteins involved in modifying cell wall? The most studied of which includes the ACTH, PG, PME, PL, and cell wall protein expressions. Different types of enzymes act on different cell wall modification, cell wall components, resulting in the reduction of cell wall integrity. Many studies found that modifying the expression of genes that encoding this enzyme influence the fruit firmness and post-harvest shelf life in many fruits. Although the link between the cell modification enzymes and the fruit firmness has been extensively demonstrated in fleshy fruits, the general regulator that regulates the expression of cell modification genes and the softening have not well been described. So transcription factors have been reported to play a role in fruit softening by regulating the expression of cell modification genes. But the most studies focus on the climacteric fruits like banana, apple, tomato, peach, and so on. These fruits had obviously respiratory climacteric and ethylene production peaks during fruit softening. So most studies generally confirm that the ethylene can promote fruit softening by regulating a transcription factor's recognition module, and the transcription factor can also regulate the biosynthesis of ethylene to mediate fruit softening. However, few studies focus on the non-climacteric fruits, especially sweet cherry. So the scientific question we want to answer is whether there are similar regulatory modules in non-climacteric fruit softening. In this study, we identified an ABA-mediated transcriptional cascade that controls the expression of cell wall modification genes and thus regulating fruit softening. So the first result is that we found the degree of cell wall degradation during fruit softening determines the fresh type of sweet cherry fruits. We firstly selected 9 cherry varieties with different firmness properties to measure fruit firmness. We at the 4 red fruit mature stage, and the variety Qingmei has a firmest fruits, while the variety Zhaodaguo has a softer fruits. Then we focused on the extreme varieties Qingmei and Zhaodaguo for further analysis. Accordingly, we collected fruits from both varieties at 8 fruit developmental stages to measure firmness. As shown in Figure 1C, with fruit ripening, firmness of both varieties gradually decreased. Notably, the firmness of Zhaodaguo decreased faster. The fruit firmness of Zhaodaguo decreased faster than Qingmei. Note we also inspected the cytological characteristics in paraffin section and the ultrastructure of cell wall disassembly using the big green and the 4 red stage fruits of the 2 varieties. Both results showed that the cell structure and the cell wall integrity is destroyed with fruit softening in 2 varieties, but the degree cell wall degradation of Zhaodaguo is more serious than Qingmei. Then we measured the changes in cell wall components across the big green, yellow, white, and 4 red stage in both varieties. We determined that the content of cellulose, hemicellulose, and covalent soluble pectin decreased in both varieties during the fruit softening, while water-soluble pectin and onyx-soluble pectin level increased over the same period. We also detected the clear differences between varieties, and the hard fresh Qingmei fruits contain more hemicellulose and covalent soluble pectin, and less water soluble pectin and onyx soluble pectin than the soft fresh of Zhaodaguo. However, the decrease in cellulose content was comparable between the 2 varieties. Collectively, these findings suggest that the fruit softening in sweet cherry depends on the degradation of cell wall, and that differences in fruit firmness between varieties are caused by alteration of hemicellulose and pectin content rather than cellulose. So after understanding the physiological basis of fruit softening in sweet cherry, we then began to focus on its molecular mechanism, and the transcriptome analysis was performed using the big green, yellow, white, and 4 red fruits from 2 varieties. The differentially expressed genes were obtained by comparisons within inter-varieties and intra-varieties in different stages, and analysis of cell wall modification genes in Qingmei and Zhaodaguo identified 21 and 23 cell wall modification genes from an inter-variety comparison, and 24 cell wall modification genes identified from an intra-variety comparison. And we also identified the transcription factors and plant hormone signaling genes that are potentially involved in fruit softening. They are co-expression analysis shows that 9 key cell wall modification genes are associated with 28 transcription factors and 27 phytohormone genes, indicating that the phytohormone-mediated transcription network plays a critical role in sweet cherry fruit softening. In recent years, ABA is considered to be the key phytohormone in promoting the ripening of non-climacteric fruits. And to investigate the role of ABA in regulating fruit softening of sweet cherry, we treated cherry fruits with exogenous ABA or its biosynthesis inhibitor NDG. ABA application accelerated the fruit ripening and significantly decreased the fruit firmness, whereas NDG resulted in the opposite effect compared to the control. In agreement with this observation, the expression of 5 keyword cell modification genes identified from the co-expression analysis were all upregulated by exogenous ABA. In addition, ABA and NDG altered endogenous ABA levels in opposite direction, promoting us to speculate that ABA biosynthesis may be affected during ripening progression. So we discovered that the expression of CYP 70782 responsible for AP catabolism decreased in response to exogenous ABA treatment, while the NCD1 responsible for ABA biosynthesis showed the opposite expression pattern. And these results suggest that cell modification leading to fruit softening in sweet cherry might be modulated by ABA-mediated transcriptional regulation. And to explore the potential transcription factors involved in ABA-mediated cell modification during softening of sweet cherry, we focused on promoters of 5 cell modification genes, all key players of fruit softening in the sweet cherry. We determined the promoter sequence and identified numerous DOF binding motifs. Then we identified 25 DOF genes named DOF1 to DOF25 in sweet cherry. We determined that DOF2, 6, and DOF15 exhibit the highest expression levels of all 25 DOF during fruit softening. And the expression of DOF6 increased with fruit development and reached its highest level as the ripening, while DOF2 and DOF15 exhibited the opposite expression pattern, and subcellular localization so that they were all located in the nucleus. Then we used the YOS1 hybrid assay and EMASA to test whether DOF2, 6, and 15 can regulate the 5 cell modification gene, and the results showed that these 3 DOFs can directly bind to the DOF binding motifs in the promoters of 5 cell modification genes. And to determine the type of regulation exerted by each DOF, we conducted a dual luciferase activity assay, and the overexpression of DOF6 strongly activated the promoter activity of these 5 genes, while DOF2 and DOF15 repressed their transcriptional activity. Collectively, these results suggest that DOF2, 6, and 15 are implicated in regulated fruit softening by directly modulating the transcription of cell modification genes in sweet cherry. Then we examined the expression of DOF upon treatment of ABA or NDG to test whether DOF participate in ABA responses, and we found that DOF2 and DOF15 expression levels are lower upon ABA treatments, while they were induced by NDG. On the contrary, ABA increased the expression levels of DOF6, but NDG had no effect. These results suggest that ABA accelerates the fruit softening partly by altering the expression of DOF2, 6, and 15, whose encoding transcription factor in turn regulate expression of cell modification genes. And to further ascertain the function of DOF, we transiently overexpress DOF2, 6, and 15 in cherry fruits. All 3 genes were upregulated in the transgenic fruits. And and a phenotypic characterization of transgenic fruit shows that fruit firmness significantly decrease in overexpressed DOF6 fruits, while it increase in the overexpressed DOF6 and DOF2 and DOF15 fruits, suggesting that DOF6 promotes fruit softening, while DOF2 and DOF15 repress fruit softening. We characterize the expression of 4 cell modification for the modification genes in transgenic fruits. These cell modification genes were all upregulated in the overexpressed DOF6 transgenic fruits, but downregulated in the overexpressed DOF2 and 15 fruits. Together, these results suggest that DOF regulates fruit softening by modulating the expression of cell modification genes. And we then measure the expression levels of AB metabolism gene in transgenic fruits. Indeed, relative ACD1 transcription levels increase in overexpressed DOF6 fruits and decrease in the overexpressed DOF2 and DOF15 fruits compared to the control, while CYP70782 shows the opposite expression pattern. And consistent with these results, we observed that AB content of overexpressed DOF6 transgenic fruits are higher, while they were lower in the DOF2 and DOF15 fruits compared to the control. And next, to test whether the DOF directly regulate AB biosynthesis, we confirmed by a method and YOS1 hybrid assay that all 3 DOFs can bind to the ACD1 promoter, and the dual luciferase active assay shows that overexpression of DOF2 and DOF15 strongly repressed the promoter activity of ACD1, while DOF6 activated its transcriptional activity. Together, this observation suggests that AB-mediated fruit softening is also subject to feedback regulation by DOF2 and DOF6 and 15. And to further identify the potential transcription factors that are located in the upstream of models that we have determined, we found many AIF binding sites in the DOF2, DOF15, and ACD1 promoters, suggesting that AIF transcription factors might participate in the softening process. And we thus identified 17 AIF genes in sweet cherry. Among them, AIF8 and AIF5 and AIF8 expression levels decreased during fruit development, and exogenous AB treatments only led to a decrease in AIF8 expression pattern, promoting us to focus on this gene. And the YOS1 hybrid assay and method confirm that AIF8 can specifically bind to the AIF binding site in the DOF2, DOF15, and ACD1 promoters. And the dual luciferase activity shows that AIF8 can increase the promoter activities of DOF2 and DOF15, but decrease the promoter activity of ACD1. And together, our molecular studies confirm that AIF8 can act as upstream of DOF2 and DOF15 and participate in the AB-mediated fruit softening. And in summary, we found that AB can promote fruit softening by increasing the expression of DOF6 and suppressing the expression of DOF2 and 15. DOF2, 6, and 15 can regulate the expression of cell modification genes by binding to their promoters to directly regulate fruit softening. And DOF2, 6, and 15 can also feedback regulate the expression of ACD1 to influence the AB biosynthesis and indirectly regulate fruit softening. And the last, AF8 acts upstream of DOF2 and 15, participating in AB-mediated fruit softening. So finally, I want to explain my acknowledgement to my supervisor, Professor Tianhong Li, and the teacher Yang Liu, and Professor Bingbing Li, and my partner, and my other partners. Okay, thank you. That's all.
Thank you so much, Zefeng. So while we're waiting for your questions, could you maybe quickly comment on the conservation of this mechanism in other non-climacteric fruits?
Sorry, I can't.
Do you think that this mechanism is conserved in other non-climacteric fruits? Do you have any?
I think that in recent years, the ABA is a key phytohormone in the non-climacteric fruit. I think such as other not just sweet cherry and other non-climacteric fruits, I think it's conserved.
Okay.
But the function of the DOF genes need to be confirmed.
Okay, so we have one question. Okay, so somebody's commenting, I'm confused about the role of ARF8 in this mechanism. Could you explain that a bit more?
This work, I don't finish this work. This just a clue for the study, and the next the ARF8 is the oxy responsive factor. I think the crosstalk between oxy and ABA is very important in the fruit ripening. Not just in non-climacteric fruit. So our next study direction is about the crosstalk between the oxy and acetic acid to coordinate regulate the fruit ripening, and specifically our AF8 in the mechanism and we another doctor is doing this work.
Yeah, yes, this is interesting. So somebody else is commenting. So auxin plays a role too. Yes.
Oxy.
Yeah.
I think it's inhibit fruit ripening in sweet cherry.
Okay, very interesting. Thank you so much, Zefeng. I don't think we have any more time, but of course you can always contact our authors and they will be happy to answer, I think. With this, I will thank again to all speakers and go back to Richard.
Hello, that was excellent. Thank you very much. Really good talks, some nice questions. I don't have anything further to say. So Mary, did you want to finish off? Apart from me saying thank you, Aida, for excellent work there, really good. And thank you for all the speakers from me. Oh, Mary, you're doing a Richard now. Sorry.
Thank you, everyone. Just a quick note that we will be having another Plant Phys webinar in September on the cell polarity, and a plant cell webinar also in September on biomolecular condensates. So you'll be seeing emails and messages going out about that later this year. Thank you again, everyone. Wonderful talks. Look forward to getting the recording posted. Thanks a lot. Bye.
Excellent. Thank you.
Bye.
Bye. Bye.
Thank you.